is an important day for NAOS. Maybe not so many of you in the room know our company well. It's true, we have been rather discreet until now, but it's about to change. Today, we are opening a new chapter. We'll begin by shining a spotlight on a very discussed, a very important topic for our society, aging. Now, we'd like this event to be interactive. We would like you to take part in the debate. So you can see behind me a number. You just can use WhatsApp, and you will have the opportunity to ask your question. And we will have 10 minutes, 15 minutes at the end of the conference to answer as many questions as we can, time permitting. So, speaking of questions, let's start with a quick question for all of you. How many in the room would like to live to be 120? Please, raise your hand. How many? No, but when I mean 120, I mean in good health, and good health. Okay, so to help you make up your mind, to convince you, I am really delighted to welcome the person behind this breakthrough discovery, along with a reporter for CNN, our media partner. So let's give a warm welcome to Professor Radman and Claire Sebastian.
Hello, Claire.
Good evening, everyone. I would like to welcome again Professor Radman, who, as many of you may know, is a leading molecular geneticist who has a career spanning more than 5 decades, I think, many continents, from his native Croatia to Brussels to Harvard, and in a sort of symmetry reminiscent of DNA, back again to Croatia, and who has spent the best part of 50 years, I think, working specifically on DNA, a DNA-centric focus. And his latest discovery really turns that on its head. You see the words, a new paradigm. So we're going to talk about that today. I want to ask you first, Professor Radman, tell us about this new discovery. How did you arrive at that point?
Well, it's true. Over 40 years, I've been working in the area of molecular repair, which is some kind of molecular microsurgery of DNA that gets damaged from sunlight, from chemicals, and from metabolism, from free radicals. And then after that many years, I thought, well, let me try something where people will be more interested, and I'm becoming more interested because I was in the meantime, I was aging, and to see the limits of life, to see how robust can a life-based, carbon-based life based on DNA, RNA, proteins, how robust can it be? And there was a champion of it known since end of Second World War, when the canned meat for soldiers was sterilized by gamma rays.
Canned meat.
It was after closing the meat, they were exposed to huge dose of gamma rays to kill all the bacteria or yeast, whatever, and yet there was a contaminant of colonies, or orange colored colonies of bacteria that then, taken and irradiated directly, not in the can, survived doses of radiation 1,000, 2,000 times higher than other bacteria and then our own cells.
Right.
So the question was, is this normal life? Yeah, it is. It has DNA just as others. It has proteins and so on. And this was the beginning of exploring why and how. How is it possible? Why others die and how these don't die at this huge doses. And what was seen, that DNA is pulverized in hundreds of little pieces, cut And then after 2-3 hours, the bacterium managed to take this DNA text all in little pieces and put it in the right order without any further mutation, without mistake. How can a bacterium be so intelligent? Who can read these sequences and say, aha, I know how it was before cutting, now I see the fragments, but I remember I will put them all in the order. It was an intellectual challenge. And it was exciting, and we published a nice paper in Nature. There was a lot of comments all over the world. But this was not most important. Most important was the simplicity of the solution of this enigma. The solution of the enigma was that we shouldn't have concentrated on DNA. It's spectacular that something out of ashes resurrects DNA. You know, I was joking at the time that we should apply for grant to Vatican, you know, the molecular basis of resurrection. But what at the end of the day, as in England say, at the end of the day, it turned out that the solution is almost banal, so simple. And that made me believe that we are on the good way. The simplicity was that DNA clearly is not protected, it's massacred, and then proteins, which do everything in life, including repairing genes and copying genes and reading genes into functions and so on, that the proteins were protected. Unlike in other species where the proteins with radiation were oxidized quickly, here 1,000, 5,000, 10,000 grays, which are monstrous doses, on the level of protein, nothing was happening. You wouldn't say that it was irradiated. And that was the secret. The secret was that if you protect function of life, which is proteins, you know, DNA is information, is the instruction to make function, to make proteins. But life is proteins, and I'll give you just quickly an example. Globule rouge, erythrocytes have no DNA, and they function 2 to 4 months. Very hard work, by the way, you know, playing with oxygen and with free radicals and so on. So a cell can be live, living, functioning without genes, as long as the proteins will last. Where we need genes is when the proteins burn out, they are sensitive, get oxidized, rusted, corroded, and they go into the garbage. We need to make fresh proteins. And there, the genes are necessary to make new proteins. So that was it. That it is banal. There was nothing new. It was simple, and I'm proud that we can today, in few minutes, explain the simplicity, unlike anybody in the world now about aging and diseases, that I can shock you mildly, that saying aging is simple, diseases is simple. What is complicated are the consequences of diseases and consequences of aging. But the cause is simple, and we can act on that cause in order to mitigate, to prevent all of the diseases and to slow down the aging. What is done now is looking for the consequences of the consequences of the consequences, and they're called biomarker. Something goes up, something goes down in aging and disease, and then you target the molecules of pharmaceutical companies, and there's no result. There zero success of it. So I'll play the bad boy later and say
we have decades of what you would call a DNA-centric approach now turned upside down, and you're looking more at the role of the proteins in replicating, repairing, reading DNA. I want to home in on one type of cell, you correct me if I'm wrong, I'm not a scientist, called Deinococcus radiodurants. Is that correct? I've been practicing. Which has become instrumental, I believe, in your research as one of the more robust organisms that can provide the inspiration for further sort of slowing down, essentially, of the aging process. Tell me more about that.
It's in the Guinness Book of Records as the most robust living organism, bacterium. And who wants to catch it can catch it while snow or rain is falling, because with balloons, it was found in vacuum, in stratosphere, totally dehydrated, desiccated, irradiated with UVC light 1,000 times more intense than at the Earth, for who knows, months, years, and then eventually it falls down with snow or rain. And what happens is that this totally dehydrated organism, irradiated to 1,000 times over deadly doses, rehydrates and finds sufficient amount of still active proteins which start recovery, rebuilding, like after the war, like Dresden, that you need somebody surviving to start now cleaning the houses and building. And this is what happens with this Deinococcus radiodurans.
And this is a single-cell organism,
yeah,
right?
It is so massacred, but it has still enough of proteins. For the cell, proteins are like people for a city. All right, and for the cell, the membrane is the skin. All right, and this is key things that we are eventually going to discuss. And so, what saved life? What saved life in the sense that this clinically dead bacterium will resurrect after rehydration and so on, is that not all proteins are dead. Right. If it were my cells or Escherichia coli, all proteins would be dead, and there would be no back to life. Yeah. Here it is possible because the Deinococcus radiodurans invested into synthesis of something that other organisms didn't invest as much. And this is the small molecular complexes which bind to bind physically, not chemically, bind to proteins, and therefore they are called chaperons, comme le chaperon, in all times. The ladies protecting the young girls from bad guys. And these chaperons then stick around the protein, and they are also antioxidant. So they don't float around and to neutralize free radicals. It's there like the protection of the president by the bodyguards, kill me instead of president. So these molecules are sacrificial molecules. Right. They are killed by oxidation, and they, in a way, quote unquote, sacrifice themselves to protect important molecules such as proteins or DNA. So Deinococcus told us this story. Right. Now we could turn to more normal organisms and creatures, and finally to people, and see, what are these molecules? What are these antioxidant chaperons? Can we try them? Would it protect our proteins? The answer is yes. There's no private property of Deinococcus that these chaperons would protect only Deinococcus' own proteins. It would protect equally well, we tested that, Anita is not here, unfortunately, she did that, that it will protect our proteins and Escherichia coli proteins equally well. Okay?
So how do we take what we've learned from Deinococcus, and how do we get those chaperones? How do we become more like this extremely robust organism?
We do have chaperones. We have plenty of different families of chaperones without which we couldn't live. Yeah. Without this, but these are protein doctors of sick proteins. Like our doctors are people like us, and they try to heal other people. So these proteins are the protein doctors for misfolded, the term in English is misfolded, but for in a vulgar way, for screwed up proteins, would say, okay, vulgar, if I want to say screwed up proteins. It's not like this, which is perfect, and then this protein is resistant to oxidation, because evolution did over 2.5 billion years since the oxygen went up due to photosynthesis, and then oxygen-free radicals evolved, and the efficient metabolism, which makes a lot of energy, but makes a lot of free radicals. So rusting, okay? So there are, we couldn't live, and even the bacterium cannot live without these protein chaperones. But there's a backup system of more primitive chemical chaperones, which look a little bit like vitamin A, beta-carotene, the double bond, single bond, like this. And then at the end, they have some other groups. And so there's this primitive system that is backup system, and Deinococcus uses this primitive system to, in an exaggerated way, in a high concentration. Yeah. And it allows Deinococcus to be the Guinness Book of Record most robust cell that we know.
So I think the question is, what are the implications of this discovery that essentially proteins can rebuild, in certain cases, damaged, broken, shattered DNA? Yes. What are the implications for health? Your book talks about the key to immortality. Is that what we're looking at here?
Yeah, yes. Let's now jump to the people. Do you agree?
Yes.
Okay. What we have learned by analyzing particular human proteins, those that are linked with predisposition to early onset of heavy diseases like Parkinson's disease and Charcot disease, amyotrophic lateral sclerosis, and so on. What we learned 15 years, I may be telling things very simply and joking a little bit, but 15 years of life, not only mine, but of about a dozen of young people, that's not a joke. And so what we learned is that the chemistry of aging, we wanted to know, well, if the chemistry of protection against radiation, which is protection against free radicals, against the rusting, is there an overlap? And the answer was yes. There is a large overlap over radiation resistance and time resistance, chronoresistance. That means living long, right? Because, again, nothing is more dangerous to life, to human life for sure, than living. Living is the most toxic activity concerning the reason that I'm saying, maybe you saw me smoking, have a cigarette there. If you smoke double of the number of cigarettes, the risk of lung cancer so will double, okay? It's a linear. The more you damage the DNA, maybe whatever, the more you are exposed to something toxic. In a linear way, proportionally, there's a risk. With time, the risk of dying, of getting all of these cardiovascular, neurodegenerative, malignant diseases goes with fifth power of time. It's not like this, the curve is like this.
But how
if you double the age from 30 to 60, the risk of dying and getting the disease goes 32 times. But if we irradiate people or force them to smoke more, then doubling the dose will double the risk.
So how do these
most
proteins
dangerous
help?
is to live. And we wanted to know the chemistry of it. And the chemistry is oxidative damage to proteins. It goes, a small worm which lives 3 weeks will, in the course of his life, will accumulate the same proportion, the same amount of oxidized proteins as we will, if somebody who lives 100 years, it will. So it's whether you live 3 weeks or 100 years, the amount of accumulation of damaged proteins is the same, all right?
So it's the damage to the proteins that causes the aging. This is your discovery, not damage to DNA or the cells. This is about cell death causing aging.
Yes.
And you're looking at protecting these proteins. This is how we take this forward.
This is because the dependence, people were obsessed, myself first, with elegance of, DNA and its information is like poetry, is whatever you want to amuse yourself. But life of DNA depends on proteins. The life of proteins, once they are synthesized, does not depend on DNA anymore. Erythrocyte, right? Only proteins, no DNA, and it works well as long as the proteins are active. Yeah. Once they are burned out, new ones cannot be synthesized because there's no gene, no instruction to make. So the chemistry of aging is simple. It's oxidation of proteins. What is complicated are the consequences of that damage. This is important. One thing I beg you to remember is that because it's a judgment of entire science in last half century, and that is that what cancer research has done is to provide a lot of knowledge about normal cells. By studying cancer, you compare with non-cancer, and you start understanding the consequences of that disease. You learn nothing about the cause or the way to go back. You cannot. So the cancer research and biomedical research is used. It's not useless. It would be useless if it didn't provide the information of the consequence of disease, and then you learn from that consequence how normal organism works.
And you
learn nothing about the curing disease. This is amazing. So the complexity of disease and aging, you should stop me when the time comes. Complexity of aging and disease is the diagnostic of the complexity of the healthy organism. Nothing more.
And I think that's where we're going to have to stop.
Yes, it's like rusting of iron. Rusting of a bicycle is the same rust as rusting of an aeroplane. But the consequences are much simpler on the bicycle, and you think that I'm making the point. The same chemistry of rusting, of oxidation, will appear with simple effects on the bicycle and very complicated effects on airplane. And we think that our diseases are incredibly complicated, and no wonder that we don't have any results because it's so complicated. No, it is not complicated. It is simple. What is complicated is the complexity of our organism. So if we go to the origin, to the root cause, and act on it, then a miracle could happen. I'm stopping now. Miracle could happen that it would be simpler and more effective at the origin of these free radicals and protein damage if we can act on it, and we can act on it, and do act on it, that it will be simpler to eliminate all of age-related diseases, cancers, Alzheimer's, and all together, then pick a single one. Then a single one. This is the message. Now I can shut up. That I wanted to share with you because it's important. Because it's an option. It's an option. Shall we eliminate all of diseases that kill us in a simple way, or shall we exhaust ourselves to fix things with disease by disease as we do?
So we end, Professor Radman, then on a hopeful note. This can be something that can not only slow down aging.
This is a possibility that I would bet on, that is not excluded, and this is sufficient that it is not excluded, because you may think it is impossible to do this, but it is not. There are 4 persons per 10,000 called centenarians.
Yeah.
Centenarians don't get any of these diseases, and therefore don't die of them at the age of 70 or 80, but die by switching off at 100, okay?
Yeah.
So this is the project, this is what we have learned, the summary of we had learned from basic science, and now Emmanuel wants to tell why
Thank
didn't
you.
you
Have to
give up the floor.
Thank
why didn't you talk about skin? But there will be others. There will be other colleagues of
Thank
mine
you very much,
talking
Professor Radman.
about specifically aging of skin, right? So
Perfect. Thank you so much, Professor Radman. We have to stop there. Distilling a 50-year career into 20 minutes.
I hope
Not
that
easy.
this last message passes, because then
Yes.
I can go home and sleep.
Thank
All right.
you.
Thank you so much.
Right.
Thank you, Miroslav, and thank you, Claire, for this fascinating interview with such a body language. So thank you. So we've already received some questions, so please keep them coming for our Q&A session at the end, please. Okay? So now, beyond the genome, we have the proteome. So these few words sum up our tonight's discussion so well. Aging, aging well, age-related diseases. I suggest to continue this deep dive into aging with Professor Louise Serpell. She's professor of biochemistry and director of Sussex Neuroscience. Her specialty is understanding the misfolding of proteins associated with Alzheimer's and how they are involved in the progression of the disease. Until recently, Professor Serpell didn't know us at NAOS. She accepted our invitation, and we thank her warmly for that. So without further ado, I let Professor Serpell enlighten you.
Hello, everybody. I'd like to just start by thanking Professor Radman for just such an amazing talk. It was really, really inspiring for me as a scientist. And also to thank the team at NAOS and Emmanuel for inviting me to talk to you today. So what I'm going to talk about today is this sort of picture that I see of what's happening during aging. So what you see here is as we age, as Professor Radman has discussed, we undergo all sorts of issues. We have our own genetics, of course, and we also have bacteria that inhabit our gut and the rest of us that affect us. But we also have to consider our exposure to the environment over time. And we also may get infections and undergo stress. And these things all affect our proteome. So they all affect the proteins that we've been talking about earlier today. And over time, damage starts to accumulate. So there are 9 correlates of aging. And what I wanted to point out, really, is this main one here, this one in purple, which is the loss of the proteome. So as we age, there is a decline in proteostasis, which is essentially how we maintain our proteome. And as we age, that starts to decline. So we're going to focus on that today. And when we talk about proteins, we need to rethink some of the things that you might have learned in school. This is the central dogma, which you may remember from school, where we go from DNA to RNA to protein. But we need to add to this because we need to consider that this protein, which is made of amino acids, will change because it needs to fold into a 3-dimensional structure. And that's absolutely essential for each protein's function. And this next slide shows you just some of the proteins that we know make up the proteome. And what I think you can appreciate, although you don't need to be able to read this, is that they're all different shapes and sizes. And each one of these has a very specific function that's associated with its shape. And so each protein needs to fold correctly. And so this is what I mean by protein folding. So this is a polypeptide chain, or a protein. Each one of those letters is an amino acid. And so you can think of it as beads on a string. And this is what it looks like when it first gets synthesized. And it needs to fold up every single time into exactly the correct functional structure. So every time a protein folds, it needs to always fold in the same way and end up looking exactly the same shape and size in order to perform its function. And so that's what's shown here. This protein is now fully folded, and every time it folds, it will always fold the same. Or will it? So sometimes proteins misfold, and I think Professor Radman did mention that. So misfolding is when the proteins don't fold correctly. This means we lose potentially their function, but we also have another problem, which is a gain of function. And what I've done here is to show you this as a movie. So essentially what this is some proteins in a box. And it's in silico, so it's in a computer. And so what you notice is that these proteins have been given a bit of energy, so they're just moving about. And I think what you might notice is as they start to move about over time, they start to associate with one another. And they don't associate in just a very random sort of way. They actually do in a very organized, ordered way. And you might be able to see here at the top that these proteins have organized themselves. They almost look stripy. So they've organized themselves in a very specific, organized way, or we think of as ordered. And this is really important. These proteins misfold, and they aggregate or they self-assemble. That's the way that we discuss them. And so it's really important that we maintain the proteome. So we've obviously evolved in order to maintain the protein. Each of our proteins, and this shows a whole scheme of what's going on in the lifetime of a protein. And although I don't have a pointer, I'll have to just sort of wave randomly, and these little wiggly lines, the red ones, go on to form this correctly folded native structure that you see on the right-hand side. They can then sometimes, during their lifetime, be degraded. Then that forms the amino acids that then are recycled to make new proteins. So that's a really important point. But occasionally, they will misfold and start to assemble to form these lower structures that are called amyloid, which I'll talk to you a bit more about. And if we have oxidative stress, which accompanies aging, then this can speed up the likelihood of that happening. And what I want to point out in this slide is these green sort of circular structures, which are chaperones. So what the chaperone system, and this generally I'm talking about protein chaperones here, they've evolved in order to protect our proteins from misfolding and protect them at all the vulnerable stages during their lifespan. So if we disrupt proteostasis, then potentially we end up with a set of diseases which are called amyloidoses. And this is a collective name for protein misfolding in disease. So it can be all sorts of proteins. I can tell you a bit more about that in a moment. Some of these are neurodegenerative diseases like Alzheimer's disease, and some of them are systemic or peripheral amyloidosis, where you might get amyloid collecting in different organs like the heart, the skin, or in the kidneys, for example. And what we notice when we look at these graphical representations of Alzheimer's disease, Parkinson's disease, and amyotrophic lateral sclerosis is we see that as we age, the likelihood of us getting these diseases increases, and it does exactly what Professor Radman said it would do, to curve and increase as we age. The likelihood increases as we age. And so what exactly is this amyloid? So we can define it as protein aggregation or assembly or accumulation in the tissues. And we can follow its trajectory. So we can follow it from beginning at the left-hand side where we have an individual protein chain which starts to self-assemble and starts to form these long fibers. And these fibers all collect together. And on the right-hand side, you can see an amyloid plaque, which is found in Alzheimer's disease, for example. This is an example of an amyloidosis where amyloid fibrils collect in the heart muscle. So on the left-hand side, you can see a normal heart muscle, and on the right-hand side, you can see a heart muscle where somebody is suffering from a light chain amyloidosis. So that's accumulation of antibodies in the tissue, and you can see that that would stiffen the heart and cause disease. And this is what these amyloid fibrils look like when you look at them at very high magnification under an electron microscope. And so that you can see that they're long, long chains of protein. And just to very quickly say that there are lots of different diseases that are associated with different proteins, all of which form amyloid, and all of them are associated with aging. Some of them are genetic as well, but even those will increase in likelihood as we age. So I want to give you an example of Alzheimer's disease, and the reason for that really is because that's what I work on, and that's particularly my area of interest. And so what you see here is on the left-hand side is a healthy brain, and on the right-hand side, an advanced Alzheimer's disease brain. And you can already see that there's neurodegeneration that's gone on the right-hand side. And so if we look into this brain tissue, we see these aggregates of proteins, and these are amyloid plaques that you can see under a light micrograph. And then if we look in a bit more detail, now you can see these amyloid fibrils. So there's these long chains of protein that have self-assembled and accumulated in the tissues. And then if we look in more detail even, we can see the proteins stacked up to form this structure. And if you remember when I was showing you that movie, this is a very organized arrangement. This is not just a random collection of proteins. This is very organized. And it's held together all the way along the fiber by hydrogen bonds, making this incredibly strong. So essentially, these misfolded proteins accumulate in the tissue, and we can't remove them. They're really rock hard. And the other thing to notice about this is a very repetitive structure, which means that lots of different proteins can access this structure. And often I talk about this a little bit like the rungs of a ladder. So each one of the rungs is a protein. So the important thing to consider is that these amyloid fibrils are not good for us. When they collect in the tissue, they're clearly not good for us. But also the process of assembly seems to cause disease. And we know that they affect different parts of the cell. And in this example, I've used a neuron or a brain cell because I've been talking about Alzheimer's disease. But this would be true of any cell, for a kidney cell or a heart cell or a skin cell. And these cells have lots of different organelles within them. The mitochondria that produces power, the lysosomes, which degrade proteins, so essentially they're the dustbins of the cell. In a neuron, we have to transport cargo from one end of a cell to another. And again, in neurons or brain cells, we need the connections that give the message to the next brain cell. And all of these are surrounded by this plasma membrane. And I think Professor Radman referred to this as the skin of the cell. So we know that each one of these things damages. So essentially what we think is happening in aging is this is a cascade event. So what happens is that initially the genetics, our genetic background and our exposure to different environmental factors will depend on how likely we are to succumb to one of these misfolding diseases. In Alzheimer's disease, this leads to a triggering of the aggregation of amyloid beta, so this is one of the proteins involved in Alzheimer's disease, which then we believe leads to the aggregation of another protein called tau, which is the next one down, and that spreads throughout the brain. So that leads to this distribution and neurodegeneration that happens in Alzheimer's disease. And as you see on the left-hand side, aging increases this likelihood, and of course lifestyle and environment will play a role. So what do we do about this? This is an image I showed you earlier on where we see the process of aggregation. And if we want to try and stop it, we need to try and stop it at some point during this pathway. Perhaps we choose one of the arrows to try and stop it at that point and prevent it from going any further. And so you may have heard in the news there's an antibody which specifically binds to a particular species in this pathway. It's called lecanemab, and it's an antibody that binds around that sort of point in the pathway. But actually, on this slide, I've also shown that green structure, which I referred to earlier as a chaperone. And if we really want to stop these diseases, what we really need to do is to prevent this misfolding pathway from happening in the first place. And the best way to do that would be to use the chaperone system. So let's go back to that slide I started with at the beginning. Now, in this time, I've added in the chaperones, because what we need to really consider is, could we change the trajectory of this proteome decline over time if we were able to harness the power of the chaperones? And what we know is that with aging, the efficiency of our chaperones declines. Many of them are proteins, so they decline too. And so what we need to do is to really try and harness the power of these proteins and other chaperones. So just to finally summarize, what I've talked to you about today is the proteome and how aging leads to damage of the proteome. This leads to protein misfolding, and then protein aggregation, so the self-assembly of these things to form these amyloid structures. And this leads to tissue degeneration and disease. So we need to try and tackle this by stopping it in its tracks. So I want to just finish by acknowledging my team. So shown in all of the pictures here are all the people that actually work with me and do the experiments in the lab. So through the lab, lots of students go through and lots of staff, and it's a really lively, exciting environment. Along the bottom and at the top, I've included some of the funding that we have from charities and also from grant agencies run by the UK government. These are all the people. And I just wanted to finish by thanking you all for listening so quietly, and also to mention this is Brighton, this is where I work and live, and occasionally it's really beautiful. So thank you very much.
Thank you. Thank you so much, Professor Serpell.
Okay.
Yeah. Thank you for these important insights on health. So what if we talked about the skin now? But I must warn you, it won't be about miracle solution or Benjamin Button secrets, just facts and conviction based on science, on scientific demonstration. So for this, I am pleased to introduce a member of our scientific committee, Isabelle Benoit. Isabelle, the floor is yours.
Good evening, everybody. I'm very happy to be here with you tonight. I would like to conclude this session with a focus on NAOS aging science contribution to the momentous step forward that we are witnessing today with this new scientific era. A focus on the R&D process that allowed us to go from the scientific theory to a solution, a tangible application for age management strategies. So if we rewind a little bit, the basis of our work was this new scientific paradigm by Professor Radman that proteome protection is the key to cellular longevity. Indeed, proteins providing all the functions of life in a cell or in a tissue, it's only logical that life extension in good health, which is our definition of longevity, it's logical that this increased longevity implies protecting the proteome. It's logical, but it's also, I would say, experimented in nature, and it has even been validated by evolution itself. Indeed, Professor Radman, this new theory that you posit today is the result of studies that you conducted on examples of evolutionary perfection. While trying to unlock the secrets of longevity, you studied longevity heroes. These extremophile organisms able to survive in environmental conditions that should kill any form of life. Extremophile organisms can sustain high level of radiations, extreme temperature changes, high level of oxidative stress, desiccation. And they are called examples of evolutionary perfection because, in a way, they manage to transform a hostile environment into an optimal one. More particularly, as Professor Radman said, he studied Deinococcus radiodurants, these small bacteria from the can of meat. Radiodurants, for those of you who took Latin, means able to sustain high level of radiation. And as Professor Radman said, these tiny bacteria can survive doses of radiation that would kill a human body. So as a stubborn geneticist, Professor Radman, when he began studying these bacteria, he was expecting to find solutions, explanation for the extreme robustness at the level of the DNA. But he was surprised to discover that it's not at the level of the DNA. The DNA gets fragmented, but it doesn't kill the bacteria because the DNA repairing proteins are efficient performing. So the key to longevity is not robustness of the genome, it is robustness of the proteome. And because resistance to radiation and resistance to time go hand in hand, this is also true for human beings. This is when the partnership with NAOS comes on stage. So this picture dates back from the early days of your encounter with the NAOS CEO and founder, Jean-Noël Thorel. Picture was taken maybe 15 years ago. Well, first let me say, Miro, that just for the sake of scientific demonstration, you didn't age. So you are the living proof of the relevance of your theory. Huh? But back to our topic. I'm talking about an encounter, but it's rather a matter of friendship, loyalty, and shared vision, a positive vision about life, a vision about how science should contribute to the society, and the will to be the change they want for the world. So with the help of NAOS, Professor Radman could elaborate further on his theory in the MEDIS, Mediterranean Institute of Life Science, and he granted the exclusive application of his work to NAOS. So application of Professor Radman for human care and skin care is for NAOS, and this is what we will describe today, what we discuss today. So this is great privilege for us, this exclusive right of application. It's also a responsibility, because if we are convinced, I should say as we are convinced, that proteome protection could be a breakthrough in age management strategies, it's our responsibility to transform the theory into a solution. And this is a scientific journey I would like to guide you through tonight, how we went from theory to application for age management strategies. Let's begin with a little bit of vocabulary, because we talk about proteome, but maybe not everybody knows what proteome is. So we can say that proteome is to the proteins what the genome is to the genes. It means that the proteome comprises, I'm lost in my, the proteome comprises all the proteins in a body, in a cell. Proteome comes from the ancient Greek protos, which means first and essential. First, because in terms of quantity, after water, proteins are the second most abundant element in a cell, in a tissue. If you take the skin, for instance, in terms of water content, depending on age, you have between 60% of 70% of water. Protein content in the skin, between 15 and 28. So if we take the extreme, 70% of water, 28% of protein, it doesn't leave much room for the rest. Okay? First. Essential. Indeed, proteins are essential to any form of life. They can be structural protein or they can be functional proteins. Structural proteins in your skin, you know this. Collagen, elastin, keratin. large molecules that constitute the scaffold of the skin. But you also have other proteins, usually smaller, involved in all the machinery of the skin, involved in all cellular functions. You have growth factors, hormones, cytokines, interleukins, enzymes. All of these are proteins, and they provide all the functions of life in the cells and in the tissues. If our skin is a living world, it is thanks to the proteome. It is via the proteome. So logically, any impairment of proteome functioning impairs the life of the skin, and damage to the proteome is the root cause of skin aging. This discovery that proteome damage is the root cause of skin aging induced us at NAOS Aging Science to completely redirect our R&D strategy. We aimed all our efforts at protecting the proteome. This is a significant change because the dogma so far was rather DNA-centric, genomic-centric. But we do believe that it is more important to protect the proteome rather than protecting the genome. Why? First, because we learn from nature. And what nature teaches us is that when the proteome is damaged, when the genome is damaged, it is the proteome that repairs it. So it's better to cure the doctor rather than cure one isolated patient. This is the first reason. The second reason is based, I would say, on common sense. The genome, as we all know, is a full set of genes. The genome represents everything that could happen. But not all genes are expressed. Actually, many of them remain silent. It's only when a gene is translated into a protein that the gene becomes reality. So to summarize, we could say that the genome is a virtuality and the proteome is a reality. So it's always better in terms of efficiency to focus on the reality rather than on the virtuality. Proteome rather than genome. Okay? So what should we protect the proteome from? The main damage to the proteome is called carbonylation. Carbonylation is a specific kind of oxidation, so it is mediated by free radicals. It is specific to proteins, and it's particularly damaging to the proteins because it blocks the protein under a form under which it is no longer functional, impossible to repair, and very difficult to eliminate. If we remember the intervention from Professor Serpell, a protein, in order to be performing, must be folded in a specific 3-day conformation. In this conformation, the sensitive parts of the protein are hidden inside the core of the protein. They are protected. When the protein unfolds, in case of stress, in case of problem of replication, it unfolds and it exposes the sensitive parts, which are called hydrophobic amino acids. So these hydrophobic amino acids that used to be protected within the core become exposed, and they are the target of attack by free radicals. And this is the carbonylation. Problem is that unfolded protein, once it is carbonylated, cannot be refolded because the carbonyl group is like a lock. Okay? So the carbonylated protein cannot be repaired. It begins to aggregate, and you have aggregates of protein in all age-related disease, as Professor Serpell told us. The carbonylated protein aggregates are markers of age and accelerators of age. Markers because you can correlate the age of the cell, the age of the tissue, with the number of protein aggregates. Okay? These protein aggregates are the hallmark of all age-related diseases. An accelerator because these aggregates are too large to be digested, eliminated by the cell natural detoxifying system, so they accelerate aging. The consequence on the skin of carbonylation can be seen on all the layers of the skin. At the level of the stratum corneum, the outer layer of the skin, carbonylation induces loss of hydration. At the level of the epidermis, carbonylation induces loss of radiance, loss of evenness. And in the dermis, increased wrinkles, less firmness, and less elasticity. So what we must keep as a take-home message from this slide is that preventing protein carbonylation ahead allows you to have, with one single approach, comprehensive approach, to have an effect on all age signs of the skin. You only prevent skin protein carbonylation, and you have an effect on the 5 signs of aging. How could we protect the proteome? If we remember, we have the protein, which is the target, and we have the attackers, the free radicals. The most efficient approach would be to protect the protein and to neutralize the free radicals. To protect the protein, we have natural effectors of protein protection in any organism, called chaperones, as Professor Serpell said. Chaperones, which are produced in normal state with a role to properly fold the protein, are overexpressed in case of stress, with the role to either refold the protein or to cover the sensitive amino acids, the parts which are sensitive to carbonylation. It's like a physical shield, a way to reduce the sensitivity of proteins to carbonylation. And we also have, in the wild, in nature, in our body, molecules that naturally scavenge the free radicals. They are called antioxidants. So the best solution to have a complete protection of the proteome would be to be chaperone-like and antioxidant. This is where NAOS aging science comes on stage, because we found this naturally occurring chaperone-like and antioxidant molecule, where in another extremophile organism, so not Deinococcus radiodurants, but the little sister, I would say, called snow bacteria. The Latin name is Arthrobacter agilis, but I prefer snow bacteria because it also shows where it was found. It was found in a snowflake. It was first isolated from a snowflake. So this is a true story. It's a genuine story. It was snowing one evening in Paris, and a researcher from the team of Professor Radman took a snowflake and tried to check whether maybe some bacteria would have survived. And he found the snow bacteria. This snow bacteria is able to sustain high level of radiation up in the sky, extreme cold, extreme dehydration. It's an extremophile organism. How can these tiny bacteria survive so aggressive environmental conditions? Because it has learned to synthesize its own proteome protectors, small molecules called bacterioruberins that snow bacteria has managed to synthesize. These bacterioruberins are at the same time chaperone-like and antioxidant. And specifically in snow bacteria, bacterioruberins are available under 6 different forms. It's a unique fact in nature. You can find bacterioruberin in other bacteria, but only in snow bacteria under 6 different forms. And these 6 forms sign a special affinity for a wide array of proteins. With 6 different bacterioruberins, you can protect a wide variety of proteins. It's a little bit as if this tiny bacteria has benefited from 3.8 billion years of evolution to be able to synthesize the ultimate protector of the proteome. And this is what we extract at NAOS Aging Science. We extract these 6 different forms of bacterioruberin. And this is a way for us to complete the circle. We have inspiration from evolution with this new paradigm of proteome protection. We have the exquisite design by nature to find these molecules that are at the same time chaperone-like and antioxidant in order to optimize skin natural resources, to optimize the proteome, to protect the proteome of the skin. With proteome protection, the skin is empowered to be the actor of its own health and beauty, longer and better. It can fulfill the mission it has been assigned by centuries of evolution to be the protector of our organism, to be our best ally to withstand the passing of time. So I repeat, with proteome protection, skin is empowered to be longer and better the actor of its own health and beauty. This is the very definition of ecobiology. Our skin is a living world, and ecobiology protects it.
Thank you, Isabelle. Thanks a lot. You stay with me?
I stay with you.
Okay, so just before the Q&A session, we have a special message from our founder, Jean-Noël Torel. Jean-Noël is a man with vision. Let's take a look.
Bonjour à tous, je suis très heureux d'être parmi vous, un peu malheureux d'être par voie interposée et vidéo, mais je tenais absolument à vous dire quelques mots. Je suis plutôt fier au fond, de mettre sur la table avec Miro, avec Isabelle, cette molécule et cette avancée scientifique par rapport à la découverte de Miro sur la protéine par rapport à l'ADN dans les processus de vieillissement. Aujourd'hui, on se limite à la peau et on peut aller beaucoup plus loin. On est déjà dans les applications dermatologiques. Et il y a un premier pas qui fera suite à cette conférence quand on présentera le produit, qui a donné des preuves un peu exceptionnelles. C'est normal par rapport à une molécule exceptionnelle qui peut prétendre peut-être demain à traiter des dégénérescences au niveau des organismes vivants. Alors, est-ce qu'on rêvera d'être un jour au niveau de Parkinson, Alzheimer? J'en sais rien. Mais si on ne rêve pas grand, on ne risque pas de découvrir des grandes choses. Je suis fier du travail de mes équipes. Merci beaucoup de votre écoute.
And remember, we need to dream big to go far. So I will ask Professor Radman and Professor Serpell to join us on stage, and I will propose you three to have a seat and answer the few questions we receive, and thank you for that. Okay, just a few minutes. First question for you, Miroslav. Okay. Are you ready? So just explain us, do chaperone molecules exist in all living beings everywhere? Is it clear for you, my question? Do chaperone molecules exist in all living beings?
I don't know.
Really?
No, I don't know. The protein chaperones are very well studied fortunately, since a long time. But the property of the chemical or small molecules, chemical chaperones, is not that advanced really. So I couldn't say for sure. What we have to do is to see how these bacterioruberins, which change the charge and polarity by varying the sugar entities at the end, how effectively they would penetrate. They become, both hydrophobic in the middle and hydrophilic at the end, and how effectively they go into the cell. Now, we have to deal now with the phase that is where you can't be too smart. You have to test and find what is pharmacokinetic of these small molecules. It's good news that they are small, because if they are big, you can't transport a whole protein into individual cells. So fortunately, they are small. But when they are small, then you have kidneys, liver, and all. So this is, for me, the boring, the more painful part of the work is getting to real application, to a product that one would want to take and oneself. Okay, and it's only if they are lucky, it could take just a few years, but otherwise it could take a long time.
Okay, thank you.
Yeah.
Okay. Yeah. Second question for you, Dr. Serpell. You are talking about neurodegeneration due to amyloid beta, sorry, I'm not a scientist, so amyloid beta and tau aggregation and spreading. What are the other age-related diseases concerned by protein misfolding and aggregation? I think you talk about that, but just remind us the other age-related disease concerned by protein misfolding and aggregation.
So there are many diseases that are associated with aging, and the ones that come to mind are things like Parkinson's disease that people will have heard of, motor neuron disease, and even some of the genetic forms of amyloidosis. They don't happen in people until they're in their 50s. So even with a genetic predisposition for a disease, it doesn't actually manifest until much later. And so that suggests that aging is having a really serious contributory factor. Diabetes type 2 is a good example of a protein aggregation, protein misfolding disease that tends to affect people later in their lives.
Okay, thank you.
It's not only neurodegenerative disease, it's also diabetes. So we really have a good correlation between protein aggregates and all age-related diseases.
Okay, and you, Isabelle, just one question. Let's check my notes. How do you position the proteome approach versus the other theory of aging? I mean, telomere shorting, for instance.
It's a very good question, because tonight we don't want you to believe that this is just a new theory of aging. We have heard various theories of aging in the past, and this is not today a new one that will be out of fashion in few decades. We believe indeed that proteome protection is the mother of all theories of aging. We talk about meta theory of aging, because proteome protection overlaps with most theories of aging. If you are talking about telomere attrition, which is one of the most famous theories of aging, so you remember telomeres, which are at the end of the DNA, with the role to protect the DNA. With successive replication of the cell, the telomere shortens. And you can correlate, so it's an inverse correlation. The shorter the telomere, the older is the cell. So to prevent telomere attrition, to prevent aging, you want to prevent telomere attrition. There is an enzyme in the body who is responsible for that, called telomerase. So you want to boost telomerase to prevent telomere attrition. But telomerase is a protein, and it is one of the most sensitive to carbonylation proteins. So we see here the overlap between proteome protection and theories of aging. And you can do the same with other theories of aging. Therefore, this notion of meta theory of aging.
Thank you. Okay, we have one last question. So if we don't answer to all your questions, we're sorry, we'll try to propose you written answer. Okay.
Feeling lonely?
How is it that a product can penetrate up to the proteome and take care of it?
So that's a question for me, I suppose?
Yes, I think so.
It's something that we have demonstrated. First, we put the bacterial ruberins in a specific galenic that allows to go through the various skin layers. So first, you have to remember that you have proteins at all skin layers. Okay? So while the active ingredient penetrates, it protects all the proteins at all the various skin levels, in all the layers. And we have demonstrated using Serpell, which is a specific test to measure the penetration. We have demonstrated that we have good penetration of our active ingredient. This is work which is conducted in NAOS aging science. Therefore, we can hope to have an effect on all aging signs.
Thank you to all of you. Just one last word from...
That from the two well-known, this family of bacteriorubberins that Isabelle mentioned, and then a manganese complex from Deinococcus radiodurants. So both small but relatively complex molecules that protect proteins against oxidative damage have been shown in either worms or in flies, in Drosophila, in fruit fly, to extend, just given as a drinking water, just give it, in the drinking water, to extend the lifespan for about 40%, which is marvelous. That means that in spite of these fears, will it penetrate, will it be cleared by, digestive and enzymes and livers and kidneys and so on? Well, leave that. There is an effect on these animals, so it says that it makes sense to be optimistic and try eventually in people. On the skin is the easiest, of course.
Yeah.
But I'll be the one first to drink.
Okay, and we have that type of beverage during the cocktail, so
yes.
Thank you.
Right.
Okay, thank you to all of you. Thank you for taking part in this NAOS Aging Science Conference. Professor Radman, Professor Serpell, and Isabelle Benoit, we really appreciate all your insights this evening. Thanks for sharing your knowledge with passion, with conviction. Thank you very much.